today. So, yeah, I've known Bethany for quite a few years. I don't remember exactly when we met. It might even have been 15 years ago, so it's been quite a long time. So yeah, I've seen Bethany at different conferences where it was most of the time around this topic of quantum biology, because that is what brought us together, quantum biology. She expressed an interest in that topic, but not so much in photosynthesis, photosynthetic light harvesting, which I had been working on, but more in other different types of systems. And yeah, during her PhD, she looked specifically at the brain and how nuclear spin might play a role in neural activation. A very interesting topic. So yeah, quantum biology, as you will hear today, is a very broad topic because it investigates a lot of different systems where quantum effects have either been proven or speculated to play a role. So in the case of photosynthesis, I would say this is the best proven example of quantum biology, especially if you consider the excitonic states, which are superposition of single molecule excitations. But Bethany has been looking at more, less studied systems, I would say, where quantum biology has received also less attention, but nevertheless very interesting. And what I like about her lectures is that she makes it always interesting with the information about the latest developments of the research field, which she expresses in a clear manner. So I'm sure that everyone is going to learn a lot about this field of quantum biology today, and I'm also looking forward to it. So Bethany, thank you for being willing to present this topic to us, and I'm looking forward to what you will say.
Thank you so much, Chart, for that lovely introduction. And thank you so much for the invitation to talk. And thank you to all of you for listening. So I think quantum biology, as Chart said, is a really fascinating field of research. And in this presentation, I'm not going to go into any detail about one topic. I want to give you some broader idea of all the various topics of interest in the field. And hopefully this will spark your interest and get you to look more deeply into quantum biology. So just a little bit about me. I'm doing a postdoc at the Quantum Science and Technology group at Stellenbosch University under Prof. Francesco Petruccioni. And we've got quite a small, I mean, this is an old photo, so not everyone is still there, but these are the quantum biologists who are working on some really interesting topics. And if you're interested in looking up more about this, then go to the QSUN website. So this is a talk on quantum biology, but what exactly quantum biology is still an open question. Is it just another iteration of biophysics, or does it deserve a category of its own? I'm not going to waste too much time on this debate, but I want to tell you why I think it is really, really interesting. So quantum biology looks at things like photons, electrons, and protons in the materials that make up living organisms, like proteins or fats or cells or even the brain. You might think that this is a terrible oversimplification of the complexity and diversity of nature, which is an argument that is often raised against quantum biology. So to start this presentation, I thought it would be interesting to see whether there are any fundamental similarities between biological systems on the microscopic scale. So on the left of the slide is a simple single-celled organism, which is a bacteria. It has a cell body, and then it has those little hairs you can see that stick out like a tail called a flagella. And this flagella it uses to respond to environmental cues and stimulus and move towards and away from things. So flagella are powered by membrane potentials, which just means the concentration of charged ions across the cell membrane. And this concentration can be changed by opening pores in the cell membrane to let ions go in and out. And by ions, I mean charged molecules like calcium or potassium or sodium. Altogether, it's a mechanism for sensing and reacting to the environment and then responding. So on the right is another single cell, but this is a nerve cell. And nerve cells, as you know, collect into groups and constitute the brain, which is maybe the most astonishing of nature's creations. But on a mechanistic level, a single neuron is not so much different to the bacteria. So in a response to a stimulus, pores called ion channels open and they change the membrane potential in exactly the same way by allowing charged ions to move across the membrane. If this potential gets high enough, the nerve fires and a signal is sent down that long tail of the nerve that's called the axon before being processed and resulting in some sort of response to the stimulus. So we might consider ourselves with our brains and our consciousness very far removed from being a bacteria, but on the microscopic level, there are some quite profound similarities. So let's look at this in a little bit more detail for a specific example of a stimulus or an environmental stimulus, which is light. So one of the distinguishing characteristics of living systems is that they are both discrete from the environment, but they also have to be open to the environment and they have to extract energy and information from the environment. And to do this, they've developed membranes to control the border between outside and inside. So for simple organisms, one of the main sources of energy is, of course, the sun. So how did they evolve to manage, use, and store the sun's energy? So one of the ways they achieve this is by turning this energy into a membrane potential. And they do this by using light to open these channels that I just told you about, these ion channels, and control the flow of these negatively or positively charged ions, such as sodium, potassium, calcium, across these membranes. So in effect, they create a potential difference across this membrane where one side is more positive and the other more negative. How exactly do they do this? They use light-sensitive molecules called chromophores. Chromophores are light-sensitive molecules that absorb in the visible wavelengths, and they are responsible for a lot of the color that you see in nature. And chromophores are also at the heart of most of the topics in quantum biology. So photosynthesis is preoccupied with the chromophore chlorophyll, which makes things green. The radical pair hypothesis of bird migration depends on cryptochrome, which is blue light absorbing. Another really important chromophore is retinal. So when retinal absorbs light, it undergoes an electron transfer, and this electron transfer is linked to a conformational change. And this conformational change opens an ion channel and allows these ions to flow through the membrane, changing the membrane potential. And one of the results of this is this potential can be used to power hairs like these flagella I've just talked about that cause the organism to swim either towards or away from the light. And as these first organisms evolved, it is thought that they began to organize retinal in one spot of the cell membrane to maximize the effects and better control these the movements of these flagellae. And this concentration of retinal chromophores on the cell is called an eye spot. And you can see them in red in the picture of the cells on the left. And the reason I think this is so amazing is because we are not so very far removed from our very, very distant evolutionary ancestors. We also have a concentration of retinal chromophores that absorb light and consequently change and cause a change in ions across membranes, in particular across nerve cell membranes, and cause nerves to fire. We call these concentrations of retinal chromophores our eyes. We use them to mediate the border between our inside and our outside. We also turn light into a signal that prompts our behavior. An even more fundamental question in the context of quantum biology, maybe, is what is life? There are a lot of theories about how life began, which I won't get into here. But to have life, you first need to have the molecules that life is made from. So in 1952, the Miller-Urey experiment showed that you could put a collection of prebiotic molecules such as water, methane, ammonium, and hydrogen into a flask and then send electricity through the flask. And what you get is the amino acids that are essential to building the proteins that sustain life. And some theories have suggested that lightning may have played the role of this electrical spark at the beginning of life. But what about in places where there was no lightning, such as deep sea vents? Were there some sort of localized sparks And there are some theories that iron sulfur compounds, such as iron pyrite, which you may have heard of as fool's gold, could have acted in this way because it is redox active and it facilitates the transfer of electrons. I really like this thought, and I've included it because if it is true, it means that fool's gold is actually infinitely more valuable than gold. But it also means that electron transfer might have been implicated in the very beginning of life. And charge transfer, the movement of electrons and protons, is still extremely important in biological systems and forms the foundation on which quantum biology is built. So before we get into the basic concepts, let's have a quick look at the history of quantum biology, which is quite closely tied to the history of quantum mechanics in general. So the origins of quantum mechanics go back to the early 20th century when classical physics could no longer explain certain experimental results. So in 1900, Planck introduced the idea that energy is quantized, and he proposed that it is emitted in discrete packets called quanta, which laid the foundation for quantum theory. Einstein built on this in 1905 by explaining the photoelectric effect using quantized light particles or photons and providing strong evidence for the particle nature of light. Then in 1913, Bohr applied quantum ideas to the atom, and he suggested that electrons orbit the nucleus in specific quantized energy levels. Compton further supported the particle theory of light in his 1923 experiment, which showed that photons could scatter off electrons like particles. In 1924, de Broglie proposed that particles, like electrons, also have wave-like properties, introducing the concept of matter waves. And this led to Schrödinger developing wave mechanics in 1926, formulating an equation that describes how quantum systems evolve over time. And around the same time, we have Heisenberg creating matrix mechanics and introducing the uncertainty principle, which states that certain pairs of properties like position and momentum cannot be precisely known at the same time. And this is a broad simplification, but together these and other scientists revolutionized our understanding of the microscopic world and laid the groundwork for modern quantum mechanics. Quantum biology, on the other hand, is often thought of as being a relatively recent field of research. But the reality is that the founding figures of quantum theory were already thinking about how quantum theory might give some insight into the natural world and living systems. Schrödinger, who is famous for his wave equation and his cat thought experiment, also wrote a famous book called What is Life? Bohr, who updated our model of the atom to include quantum mechanics, gave a lecture called Light and Life. Light was central to the development of quantum mechanics. So the first ideas of quantum mechanics were the result of Planck trying to explain black body radiation and Einstein trying to explain the photoelectric effect. Both of these deal with the interaction of light with matter. Biological systems are also made up of matter, and light is fundamental to life, most obviously in the fact that plants turn light into biomass. As Chart mentioned, they photosynthesize. Schrödinger's 1944 book, What is Life, and Bohr's 1932 lecture, Light and Life, both explore how principles of quantum mechanics might apply to living systems. In What is Life, Schrödinger wrote about the molecular basis of heredity, and he was intrigued by how the order and stability of genetic inheritance could arise from the disorder of thermodynamics. And he suggested that genetic information must be stored in something like an aperiodic crystal. And this was a term that actually inspired future discoveries of the DNA double helix. And would later influence the fields of molecular biology and information theory. In his Light and Life lecture, Niels Bohr questioned whether it would be possible to describe living processes purely in terms of physics and chemistry. And the story goes that the physicist Max Delbruck was in the audience when Bohr delivered this lecture, and it inspired him to switch from physics to biology. And Delbruck went on to pioneer molecular biology. What I also find really interesting is that Bohr gave this lecture at an international congress, not on light, but on light therapy. And I'm not sure how many of you know that the 1903 Nobel Prize in Medicine was actually awarded to Niels Ryberg Finsen for his use of light therapy to heal disease, in particular lupus vulgaris. And yet, despite this very long history of interest in the interaction of light with living matter, quantum biology is still considered by many to be a new field of research. So let's look a little bit more closely at some of the main events in the history of quantum biology. So before he gave his Light and Life lecture, Bohr also delivered a lecture called The Quantum of Action and Description of Nature at the University of Copenhagen, which explored the nature of quantum mechanics and its implications for the description of the physical world. In 1932, Pascal Jordan published the first paper in which quantum biology is referred to by that term. And then in 1941, he published the book Physics and the Secret of Organic Life, in which he questioned whether the laws of quantum physics were fundamental to life processes. This book was followed shortly in 1944 by Schrödinger's What is Life, which I've already told you about. In 1963, Loudon first hypothesized that quantum tunneling might be a mechanism for DNA point mutations. And in 1966, we have our first experimental verification of quantum effects in biology when Volt and Chance observed tunneling in enzymes, which in 1974 was developed by Hopfield into a theoretical context. While it's not on the timeline, I would also like to mention the development of spin chemistry in the 60s and 70s, which is a field of research that looks at how magnetic fields could influence chemical reactions. And from spin chemistry, we get the radical pair mechanism, which Schulten then hypothesized in 1976 could be the mechanism by which birds navigate using the Earth's magnetic field. So birds were actually first observed to use the geomagnetic field in the 70s by the Welch crows. In 1989, Judith Klinman's Berkeley group first observed proton tunneling in enzymes. And then in 2000, Torsten Ritz suggested that cryptochrome could be the site of the radical pair mechanism of bird magnetoreception. And then in the 2000s, experiments also began to show evidence for some sort of quantum coherence in energy and charge transfer in photosynthesis, though whether this was excitonic or vibronic coherence is still contested. This is not an exhaustive list of the progress of quantum biology, which has accelerated in the last decade or so, but I wanted to give you some idea of the long history that it has, despite it often being referred to as a new field. So in this lecture, which is an introduction to quantum biology, I thought it would be useful to quickly go over some of the basic concepts and terminology of quantum biology. Because it is an interdisciplinary field that marries physics and biology, it can be quite challenging both for physicists and biologists who've never encountered some certain ideas before. I don't have enough time to go into anything in much detail, but I thought it would be useful to discuss the basic ideas that are relevant to both the quantum and biology side of things. So in quantum mechanics, one of the things you will hear over and over again is this idea of the quantum system. So what exactly is a quantum system? Well, defined formally, a quantum system is any physical system that is governed by the laws of quantum mechanics, which is the branch of physics that describes the behavior of matter and energy at the smallest scales, typically at the level of atoms, electrons, photons, and other subatomic particles. More intuitively, maybe, you can think of the quantum system as the thing that you are interested in looking at. So, for example, if any of you did physics at school, you probably did many worked examples on the movements of blocks down slopes or cars accelerating along roads. In these questions, the system was the block or the car. Quantum systems are just smaller things of interest, such as photons, phonons, protons, neutrons, or electrons. And for those of you not familiar with what these things are, I'll briefly give you some context. So one of the first major accomplishments of quantum theory was Bohr's model of the atom, developed in 1913. And in this model, electrons orbit the nucleus, which is made up of positively charged protons and neutral neutrons in specific fixed energy levels or shells, rather than spiraling inward, which is what classical physics would suggest. And Bohr suggested that electrons could jump between energy levels by absorbing or emitting a quantum of energy or photon. And the emission or absorption of photons or light explains the discrete lines that are seen in atomic spectra. And atomic spectra are like the light equivalent of a fingerprint for a different type of matter. Why is this important to quantum biology? Well, it tells us the fundamental way in which light interacts with matter. And you will see that this is a very big part of quantum biology. It also tells us about the movement of electrons, which is arguably at the very heart of living systems. And the Nobel Prize winner Albert Szent-Györgyi once wrote that life is nothing but an electron looking for a place to rest. So if quantum systems are electrons and protons and photons and atoms, then what are biological systems? What is the unit of biology? So in biology, a cell is the basic structural and functional unit of all living organisms. It's the smallest unit of life that can carry out all the processes necessary for life, including growth, reproduction, and energy conversion. Cells come in two main types: prokaryotic cells, which lack a nucleus, like bacteria, and eukaryotic cells, which have a nucleus and other specialized structures called organelles. Key components of a typical cell include the cell membrane, which controls what enters and leaves the cell, cytoplasm, which is a jelly-like fluid where reactions occur, and the nucleus, which stores genetic information. And some cells, like plant cells, also contain chloroplasts for photosynthesis and a cell wall for extra support. And all life, from single-celled organisms to complex multicellular creatures, is built from cells. Within multicellular organisms, cells can also be highly specialized. For example, nerve cells transmit electrical signals, muscle cells contract to enable movement. One really important type of cell is the stem cell, which is undifferentiated and has the unique ability to divide and develop into various specialized cell types. Other cell types include blood cells, epithelial cells, and immune cells, and each play a vital role in maintaining the health and function of the organisms. In multicellular organisms like humans and animals and plants, cells group together to form tissues, which then combine to form organs like hearts and lungs, or leaves in plants. Organs work together in organ systems like the digestive system or the circulatory system to carry out complex functions. And altogether, these make up whole organisms. And finally, in our concept section, what are quantum effects? And more importantly, what are these effects in biology? So a recurring topic in the effort to define what quantum biology is this distinction made between trivial and non-trivial quantum effects. And if you have followed quantum biology at all, you may have heard people talking, or more likely arguing, about whether you can even make this distinction. So for my purposes, I take trivial quantum effects to mean those effects that apply to all matter, whether it's living or non-living. So all atoms are described by the laws of quantum theory. Understanding this does not necessarily tell us anything new about living systems. On the other hand, as I understand it, non-trivial quantum effects are those effects that are implicated in the functionality of biological systems. So this could be something as simple as adding a neutron to an atomic nucleus, such as lithium, to form a lithium isotope. And this has an effect, a pivotal effect on consciousness. So the triviality doesn't refer to the quantum effect as much as the biological outcome. Examples of quantum effects that play a non-trivial role in biological function are things like superposition, coherence, tunneling, and entanglement, which you will hear more about quite soon. So now that I've mentioned some of the quantum effects of interest, let us look at the different biological contexts in which these may occur. So to make it clear just how the quantum and biological fit together, I'll focus on a specific quantum effect before outlining all of the different biological functions that it may be implicated in. So, for example, quantum coherence in energy and charge transfer has a number of different biological contexts, including photosynthesis, microtubule or mitochondrial function. The radical pair mechanism, which is an example of spin chemistry, was first developed in quantum biology as an explanation for avian magnetoreception, where the spin states of two paired electrons allow birds to navigate using the Earth's magnetic field. But there's now growing interest in radical pairs in many other contexts, such as reactive oxygen species, which cause inflammation and signaling in biological systems. And there's also the suggestion that nuclear spin pairs might play a role in the functioning of the brain. Quantum tunneling also appears to be important in the context of biological processes. And actually, historically, tunneling was the first experimental effect to be observed, and was first observed in enzymes and has been shown to contribute to significant rate reaction speedups. Tunneling has also been applied to the biological context of receptor activation. For example, olfactory receptors, which are your sense of smell receptors, have been suggested to use tunneling, which is facilitated by the vibrations of specific small molecules. There has also been some attempts to apply vibration-assisted tunneling to the action of neurotransmitters, such as serotonin, adenosine, and histamine. And in our group, we have also tried to apply the mechanism to the spike protein binding in the coronavirus. So this is just an overview of how the presentation will proceed. I will now go into more details about how each of these processes work and what research is being done into them. So first up is coherence, specifically coherent transfer processes. And by transfer, I mean the movement of some quantum system, such as an electron, through a biological material. What do we mean by coherence? So to talk about coherence, I need to introduce what superposition is. The superposition principle is arguably at the very heart of quantum mechanics. And many of you might be familiar with it from Schrödinger's famous and contested cat thought experiment, where a cat is placed in a sealed box with a radioactive atom and a vial of poison that breaks if the atom decays and then kills the cat. So the radioactive atom exists in a superposition of decayed and not decayed states until observed. And since the cat's fate is tied to the atom, it is thus said that the cat is also in a superposition. The cat is both alive and dead until someone opens the box and observes it. So don't worry too much about cats, because while it may seem counterintuitive when applied to classical systems such as cats, superposition follows very naturally from Schrödinger's wave equation, because linear combinations of solutions to the Schrödinger equation are also solutions to the Schrödinger equation. And superposition is well described in the context of superimposed waves and results in interference effects, which depend on how coherent the waves are. In the quantum context, coherence just describes the relationship between states in a superposition. So in the context of biology, we are interested in the coherence between superposed transfer pathways involving networks of chromophores. And as I have already mentioned, chromophores are these light reactive molecules found in proteins, and they are molecules that facilitate electrons moving from lower to higher energy levels. So first up on our list of possible biological contexts in which coherent transfer processes might happen is photosynthesis. So what is photosynthesis? I'm sure many of you know, so I hope I'm not telling you what you already know, but photosynthesis is the process by which plants, algae, and some bacteria convert light energy into chemical energy, which is stored as glucose. The process mostly takes place in the chloroplasts of plant cells and uses the chromophore chlorophyll, which is what gives plants their green color. Plants harvest light, which is transferred through a network of these chlorophyll molecules to a reaction center. And before I get into too much detail, for our purposes, let's simplify this to a quantum system moving through a biological material. So there is more than one quantum particle in the mix here. There's light, otherwise known as photons, coming from the sun. And then there are electrons that these photons excite in the chlorophyll molecules in the plant cell. The photon transfers its energy to an electron in the chlorophyll, causing the electron to move to an excited state. If this electron passes energy onto another chlorophyll's electron, there's an energy transfer. In quantum biology, we are interested in whether the energy is shared or delocalized over sets of chromophores, rather than moving from one chromophore to another one by one. So in the diagram, the chromophores are the molecules shown in that black circle, and each different colored smaller circle represents those chromophores that share the energy, whereas between colored circles, the energy is transferred classically. By using more than one path at once, does photosynthesis employ quantum effects to get the job done better? What does the research say? So there was a great deal of excitement in quantum biology circles when the paper on the left came out in 2007. It extended two-dimensional electronic spectroscopy investigations of the Fenner-Matthews bacteria chlorophyll complex, which sounds very complex, but is just a complex found in green sulfur bacteria that acts as an energy conducting wire, which connects light harvesting antenna to the reaction center in this process of photosynthesis. The paper claimed that its results showed long-lived electronic quantum coherence that was playing an important part in energy transfer processes. And the authors suggested that the wave-like characteristic of the energy transfer within the photosynthetic complex might explain its extreme efficiency, and that it allows the complexes to sample vast areas of phase space to find the most efficient path. So there have been many repeat experiments done to try to find the signature of quantum coherence in photosynthesis. And more recently, the consensus seems to be is that what is being observed in two-dimensional spectroscopic experiments actually involves the vibrational coherence rather than excitonic coherence. And this quote on the slide is from a recent review, which reviewed all the evidence and concluded that the inter-exciton coherences are too short-lived to have any functional significance in photosynthetic energy transfer. And as, if you remember, I said non-trivial quantum effects are quantum effects that have some sort of functional relevance in biology. Instead, they suggest in this review that the long-lived coherences originate from impulsively excited vibrations in the protein environment, so the vibrations of the protein scaffold. And the authors conclude that this is an example of nature using decoherence, because normally the vibrations of the environment would destroy the quantum effects, but in this review they conclude that nature uses the decoherence or dissipation to tune quantum processes such as energy transfer. So next up on our list of possible biological contexts for coherent transfer are microtubules. Microtubules are formed by the polymerization of tubulin dimers, and tubulin dimers consist of alpha and beta tubulin proteins. Tubulin dimers form longitudinal protofilaments called microtubules. And microtubules are an incredibly important part of the cytoskeleton of cells, and they contribute to cell shape and structure, as well as a variety of other functions. They are integral to cell division. They form what is called the spindle apparatus, and the spindle apparatus is fundamental to the division of chromosomes when a cell divides into daughter cells. Microtubules also act as tracks along which motor proteins move cellular constituents within the cell. So why are microtubules of interest in quantum biology? Well, once again, there's a chromophore at the heart of matter. This time not chlorophyll, but tryptophan. So tubulin proteins have a number of tryptophan molecules in them, and thus microtubules consist of these underlying networks of chromophores that might support coherent energy transfer. What does the research say again? Well, research into coherent energy transfer in microtubules began with theoretical modeling because the geometry and the dipolar properties of tryptophan aromatic molecules are similar to those found in photosynthetic complexes. So scientists thought, well, if they are similar in structure, maybe they're similar in function. And papers such as the one on the left involved computational investigations of energy transfer between these tryptophans in tubulin proteins, as well as some discussion of what this biological functionality might be, with suggestions that this was a quantum mechanism of signal transduction or signal propagation. There was also some suggestion that disrupting this signal propagation by drugs that bind to tubulin proteins and microtubules, such as anesthetics, were what causes unconsciousness in anesthetics. More recently, some of these theoretical suggestions have actually been verified experimentally. So in the recent paper on the right, electronic energy migration in microtubules actually made the cover of the journal. It was and it was investigated whether the repeating arrangements of tubulin dimers, which form a crystalline order in a microtubule, with lattice constants, which are short enough to allow energy transfer between amino acid chromophores, might actually transfer excitation energy along the microtubule like a natural or artificial light harvesting system. The authors used tryptophan autofluorescence lifetimes to look at this energy hopping between tryptophans and tubulin and microtubules. And they showed that electronic energy can actually diffuse over 6.6 nanometers in microtubules, and that energy transport that is explained by conventional Forster theory does not actually sufficiently explain these observations. Even more interestingly, from our point of view, is that they also showed that the presence of certain anesthetics reduce this exciton diffusion. Tryptophan excitations and microtubules led to another recent cover story, this time on a related topic called superradiance. Superradiance is a phenomenon that follows from collective excitations. So rather than one photon transferring its energy to one electron, one photon can excite many electrons at the same time. So networks of tryptophan chromophores and microtubules form very diverse architectures in many different biological contexts, such as transmembrane proteins, cytoskeletal filaments, subneuronal elements, photoreceptor complexes, and even viruses. And the authors of this paper use theoretical modeling to show the formation of these strongly superradiant states due to collective interactions among organized arrangements of tryptophan molecules in these different architectures, and showed that it leads to an enhancement of the fluorescence quantum yield. This theoretical modeling was then confirmed by experiments. So interestingly, in a follow-up paper, these results were applied to the specific context of Alzheimer's disease. So amyloid fibrils are fibrous protein structures in the brain that are associated with Alzheimer's disease and dementia, as well as a number of neurodegenerative disorders. And the authors of this paper showed that tryptophan networks and amyloid fibrils have an even stronger superradiant effect than any of the previously examined proteins. because these amyloid fibrils have very dense tryptophan molecules arranged in specific patterns. In fact, the predicted superradiant enhancement for amyloid fibrils is enormous. It's up to 5 times the quantum yield of individual tryptophan molecules. Why is this interesting? Well, the authors suggest that the presence of amyloid is actually a reaction to oxidative stress. And oxidative stress contributes to neurodegeneration through the production of free radicals, which we'll hear more about later as well. Free radicals can emit damaging high energy UV photons. And single photon superradiance means that this network of tryptophans can very efficiently absorb these high energy photons and down convert them to lower energies. And this could have profound implications for understanding and treatment of neurodegenerative diseases such as Alzheimer's. What other related biological phenomena might use quantum transfer processes in addition to photosynthesis and microtubules? Well, photosynthesis takes place in chloroplasts, which, as I mentioned in my explanation of the cell, show similarities to mitochondria. Most specifically, they both have electron transport chains. And electron transport chains are a series of protein complexes and electron carriers that are embedded in membranes, and they play a central role in cellular respiration as well as photosynthesis. And electron transport chains play a key role in quantum biology research for a number of reasons, because they combine electron transfer as well as the production of radical pairs and reactive oxygen species, all of which are topics of interest in quantum biology. So speaking of radical pairs, takes us to our next quantum effect of interest. And in this section, we will focus on spin biology and whether quantum entanglement might be implicated in this. First of all, what do I mean by spin biology? So spin, for those of you who don't know, is the property of quantum particles that makes them behave like tiny magnets in a magnetic field. When two molecules bond and share electrons, these electrons have to be in a specific spin configuration. And this means that spin can actually have an effect on chemical reactions. I mentioned that the timeline of quantum biology was lacking an entry for spin chemistry, which was a field of research developed in the 1960s and 70s to explain the effects that magnetic fields can have on chemical reactions. Spin biology is an extrapolation of spin chemistry that looks at the role of magnetic fields on biochemistry. The quantum to biology process can be summarized very briefly as the following. So for the radical pair mechanism, a photon excites an electron. This gives rise to a spin correlated but spatially separated electron pair. This initial singlet state undergoes singlet-triplet oscillation due to the Earth's magnetic field and the hyperfine interaction. This is the quantum step. So then different spin states give rise to different chemical products. This is the chemistry step. And then these different chemicals might exert a different signaling effect in the biological system, maybe translating into magnetoreception or the effects of radical reactive oxygen species in many different contexts. And that is the biology step. What does entanglement have to do with this? So quantum entanglement, which Einstein apparently called spooky action at a distance, is a fundamental phenomenon in which the quantum states of two or more particles become linked such that the state of each particle cannot be described independently of the others, even when separated by large distances. So measurements on one particle instantaneously affect the outcome probabilities of measurements on the other in a way that does not correspond to classical expectations. How does entanglement show up in the radical pair mechanism, which I've just introduced? Well, for a pair of spins, so for two spins, there are 4 possible quantum states, or 4 different ways in which the spins can be arranged relative to each other. One is called a singlet state, and then there are 3 triplet states. And it turns out that the singlet and one of the 3 triplets are in an entangled state. However, I want to be very clear that entanglement is not strictly necessary to spin chemistry. Spin chemistry just looks at how singlets or triplets form different chemical products. And it is still actually a point of debate as to whether entanglement is used in the different biological manifestations of the radical pair mechanism. So what biological functions does the radical pair mechanism play a role in? Well, the most well-known application of the radical pair in the biological context is in bird migration. The theory goes, as I've briefly explained, is that light activates a molecule in the bird's eye, causing an electron to be transferred. This forms a pair of electrons whose spins are arranged in a specific alignment called a singlet state. This initial spin state is then modulated through the interactions of the electron spins with their environment. And there are a number of different interactions that can take place to drive this modulation, such as the Zeeman or the hyperfine interaction. The Zeeman effect is the interaction of electron spin with a static external field, such as the geomagnetic field. And the hyperfine effect is the interaction of electron spin with the magnetic moments of the surrounding nuclei in the protein environment. And these interactions result in the radical pair oscillating between singlet and triplet states. And the final step of the radical pair mechanism is the conversion of the spin character into a measurable chemical outcome. Chemical binding is restricted by the spin state, which means that a magnetic field can change the outcome of a chemical reaction by changing the spin states of the intermediate radical pair. Thus, the ratio of singlet to triplet states can alter the yields of chemical reactions. And because singlet and triplet states give different chemical products, this means the Earth's magnetic field can produce different chemical products in birds' eyes and potentially let them see the Earth's field, which allows them to navigate. There have been many papers written about this from many different angles. And to give you some idea of the breadth of research, I've chosen to highlight 2 experimental papers from the point of view of biology and then physics to see what verification of this radical pair hypothesis exists. So on the left is a paper focused on cryptochrome. I've mentioned cryptochrome before, but cryptochrome is a protein found in the eyes of birds that has been suggested as the candidate molecule in which the electrons and nuclei of the radical pair are situated. One of the approaches to verifying that birds use a radical pair mechanism to navigate involves genetically modifying cryptochrome and looking at cryptochrome mutants to check whether this protein is involved in magnetic sensitivity. And the authors of this paper show that the photochemistry of cryptochrome 4 from night migratory European robins is magnetically sensitive in vitro, and it is in fact more sensitive than cryptochrome 4 from 2 non-migratory bird species, such as chickens and pigeons. So showing that cryptochrome is magnetically sensitive doesn't necessarily prove that a radical pair mechanism is at play. And one of the criticisms of the radical pair model of magnetoreception is that there isn't a direct physical proof. And the paper on the right goes some way to providing this proof, where the authors use a custom-built microscope to show that autofluorescence in flavin molecules, which are the molecules hypothesized to be involved in cryptochrome radical pair production, is magnetic field sensitive due to the formation and electron spin selective recombination of spin correlated radical pairs. But what other biological functions might use radical pair chemistry? Well, there is some suggestion that in addition to radical pairs formed from FAD and tryptophan, which are the radical pairs in cryptochrome, radical pairs can also form with oxygen radicals, forming reactive oxygen species. And reactive oxygen species are fundamental signaling mechanisms central to biological systems. At controlled levels, reactive oxygen species, such as often known as ROS, regulate signaling pathways that are involved in inflammation, but excessive ROS production can damage DNA, proteins, and lipids and contribute to chronic inflammation, diseases such as cancer, cardiovascular disease, and neurodegeneration. So although bird migration has dominated quantum biology as far as spin biology is concerned, in the last few years, there's been a real upsurge in interest in the role of the radical pair mechanism in reactive oxygen species. And this, in my opinion, widens the context and the relevance of quantum biology quite dramatically, because humans don't necessarily need a magnetic compass, but our health is highly reliant on reactive oxygen species. And what is particularly interesting to me is the emerging evidence that magnetic field dependence on ROS can mediate stem cell growth and stem cell differentiation. And the authors of the paper on the left showed in the context of planaria regeneration that weak magnetic fields change stem cell proliferation by changing reactive oxygen species accumulation and downstream heat shock protein expression. What I think is even more interesting is that different magnetic fields have different effects, and these aren't super strong fields. They're around the strength actually of a fridge magnet. And these weak fields can either increase or decrease new tissue formation in vivo. And the authors suggest that this could be harnessed as a potential therapeutic tool to manipulate mitosis or cell division. So interest in magnetic fields in biology has grown enormously in the last few years. This is an excellent review paper of the various biological contexts in which they might be found. The table is only part of the table in this paper, and I encourage you to have a look at this paper if you are interested in magnetic fields and effects in biology. I'm not going to go into great detail about chirality and the cis effect, but it is a very interesting field of research and a growing field of quantum biology. And it is worth mentioning in the context of spin biology. But I am slowly running out of time, so I'm not going to go into that much detail on this effect. And moving on to my last example in the spin biology section, we're no longer looking at electron pairs, but at pairs of nuclear spins. So this is not the radical pair mechanism, but it follows some of the same logic. Remember that spin is the property of quantum particles that makes them react to a magnetic field. Electrons have spin, but so do nuclei. And if you remember the model of the atom, nuclei are the protons and neutrons at the center of the atom with the electrons and the energy levels around them. In this example of spin biology, we are looking at spin pairs of phosphorus nuclei. And in many ways, nuclear spin is a better option for quantum effects as it is less sensitive to decoherence. In fact, nuclear spin is the qubit of choice for some models of quantum computing, precisely because of these long decoherence times or long coherence times. And a recent theory for quantum cognition suggests that brains use phosphorus nuclear spin in calcium phosphate molecules called partner molecules to entangle nerve activity. But I don't have time to go into great detail here. But if you are interested, these are some recent papers that look at the effects of these partner molecules in neural entanglement. And finally, on the topic of spin biology, I thought it would be interesting to quickly mention new directions that quantum biology has gone in. So space travel and settlement has always been fascinated mankind. And lately, the space race has been ramping up with every billionaire showcasing their new rockets. But while lots of research has been done on getting bodies into space, less research is being done on keeping these bodies healthy. The main focus seems to be on radiation and microgravity, but quantum biology can contribute another topic to this by examining what effects weak magnetic fields can have on biological systems. So, for example, what happens when we try to live on planets such as Mars, which have very weak to no magnetic fields. And a recent preprint showed varied morphological evidence that frog embryo development in hypermagnetic, so very, very low magnetic fields is accelerated. And the data imply that tadpole physiology can sense and react to the absence of the Earth's magnetic field. So I'm very close to running out of time. So I'm just going to go over our last quantum effect in biology, and I'll go over this fairly quickly. We've looked at spin biology, but the last quantum effect we'll look at is tunneling in different biological contexts. So tunneling is a phenomenon in quantum mechanics where a particle can pass through a potential energy barrier, even if it doesn't have enough energy to overcome it classically. This happens because particles in quantum theory are described by wave functions, which can extend into and beyond classically forbidden regions. Quantum tunneling is essential in many physical processes, such as nuclear fusion in stars, alpha decay, and in many modern technologies, such as scanning tunneling microscopes. And our first biological function that may use tunneling is olfaction, or the sense of smell. In this example, the quantum particles doing the tunneling are electrons, and tunneling was actually the first observed example of quantum effects. Many biological interactions involve a receptor and a molecule that binds to and activates a receptor called a ligand. And at the risk of simplification, the lock and key mechanism of binding and activation of the receptor molecule by an appropriate ligand is pretty much used to describe most biological interactions. The action of enzymes has been partly attributed to the lock and key mechanism, as well as the binding of smell receptors and neuroreceptors, which are common receptors known as G protein coupled receptors, or GPCRs. The lock and key mechanism suggests that the shape of a ligand must match the shape of a receptor in order for it to activate the receptor, a bit like a puzzle piece. But an alternative model for olfaction, or the sense of smell, has been suggested, and this is the vibrational theory of olfaction, which was formalized by Luca Turin. And it suggests that the vibrational spectrum of a ligand enables an electron transfer to take place, activating the receptor. What evidence is there for this? Well, the standard experiment for testing the theory is by selective deuteration, which means replacing hydrogen with its isotope deuterium. Because deuterium is heavier, it has a different vibrational spectrum and should then smell differently. This experimental approach has had successes and failures, with some animals able to sense or smell the deuterated odorants, and some not able to smell them. So there definitely needs to be more experimental evidence done to verify this theory. And a second biological function that may use electron tunneling is nerve activation, or the binding of neurotransmitters to receptors on nerve cells. Neural communication is mediated by the release of neurotransmitters, and when an action potential propagating along a neuron reaches the axon terminal, it triggers the release of neurotransmitters into a gap between two nerves. These neurotransmitters diffuse across this gap, called the synaptic cleft, and they bind to special receptors on the next nerve and open the ion channels and cause the next nerve to pass along the signal. And the majority of receptors that bind to neurotransmitters are GPCRs, like olfactory or smell receptors. So it makes sense that researchers have applied tunneling theories to these receptors. What success have they had with this application? There's some theoretical evidence focused on the neurotransmitter serotonin, which suggests theoretically it might use vibration-assisted tunneling. However, this is not backed up by experimental evidence, although there has only been a single experiment done to try and verify this. And then there are a few theoretical applications of vibrational spectra to classify agonists and antagonists for other neurotransmitters or neuroreceptors, such as histamine and adenosine. Then some other contexts in which electron tunneling might be important in biology. Well, we tried to apply the theory to the binding of the spike protein in the coronavirus. It is, yes. Sorry, I've worrying that I've gone a bit over time. So let me just finish this slide and then I will wrap it up. So angiotensin converting enzyme 2, or ACE2, is the receptor that binds with and modulates angiotensin proteins, but it is maybe more notorious as being one of the main ways in which the novel coronavirus invades a host cell. And the coronavirus spike protein has been reported to bind with transmembrane ACE2 receptors before invading the host cell. might this also employ some version of vibration-assisted electron tunneling? If you would like to find out more about it, we have written a paper about it, the quantum tunneling in the context of the SARS-CoV-2 infection. Even more broadly, might electron transfer in receptors be a new way to classify the efficacy and potency of receptor-specific ligands, especially as receptors have the necessary molecules for electron transfer, such as chromophores or aromatic residues. And this would have many implications, not least in the field of drug discovery. And I think I'm over time, so I'm going to stop there. And if you have any questions, please do ask me now if we have time, or email me.
Yes, thank you very much, Bethany. That was really fascinating. You've covered a broad range of topics and biological systems.
I'm not quite got to all of it. I need to speak a bit faster next time.
Yeah, there's really a lot of ground to cover. But yeah, you've given a very nice overview of the broad scope of this topic. Thanks a lot for that. Sakazi, please go ahead.
Yes. Thanks so much for that. Was a super excellent talk. I know absolutely zero about quantum biology. At least I didn't know an hour ago, but I know everything now. You get a great overview. I want to ask you though. I was looking at that diagram of the history, and I know that the Nobel laureate Ahmed Zerual was for a while, in his latter career, in addition to inventing femtochemistry, he took those techniques and opened a physical biology center at Caltech where they were using femtochemistry. And I was just wondering if he should be on that chart because what he's done is really transformative. It's not this one.
There should be. I think
just the previous one, the previous slide that you had.
The quantum mechanics one.
Yeah, this one. This way, yeah. You went further though. Or was that the last one? Okay, well, maybe I'm wrong. Maybe it's the one you had a second ago. But anyway, I just wondered, did he fit into your quantum biology definition? Because he was like a physicist, biologist, chemist. He did everything. Are you there? I think she's frozen.
Bethany, are you still there?
Hi, sorry, did I disappear?
You froze.
Sorry.
You were nervous.
I would love to. I will look him up and I will incorporate him into my next talk. Thank you so much.
Thank you.
Thank you. There's another question by Henk Brits. Please go ahead, Henk.
Hello, can you hear me?
Yes, I can.
Okay, great. I was wondering, I've been looking at consciousness just out of curiosity, and I saw that Roger Penrose implemented some ideas from Gödel incompleteness to explain a competitive advantage that consciousness gives multicellular life forms. So he basically says that Gödel incompleteness says that there's some mathematical truths that can't be proven, and that computational mathematics is somewhat limited, and consciousness kind of circumvents that. I was wondering if you have any ideas or opinions on that perspective.
I'm not that familiar with the Gödel Penrose stuff. I'm more familiar with his microtubules and quantum gravity, so orchestrated objective reduction. And honestly, I think I'm more interested in the sort of mechanistic level for quantum effects I mean, we don't even understand the neural correlates of consciousness. So making claims about consciousness before we even understand the machinery or the of consciousness, for me, is a step too far. So I'm more interested in how do quantum effects allow nerves to work, and if nerves are what give us consciousness, then that's a first step. Let's see how quantum effects affect ion channels or affect neural firing or affect the brain, and then maybe we can say something about consciousness. I mean, that also, I mean, maybe nerves are not necessarily the only cell that can give us consciousness, because I know that plants respond to anesthetics. So I do think with consciousness research, we haven't defined exactly what we mean by all the terms. What is consciousness? What is self-consciousness? What is awareness? What is sentience? So I think consciousness needs a lot more tightening up as a science before we make any claims about it. I don't know if that answered your question. I'm not familiar with the Gödel theory, so I have to say I'm wary of quantum claims of consciousness before we even understand quantum mechanisms and nerves.
Yes, that's fair. It was just, I think, an explanation for why natural selection selected for consciousness.
Okay, interesting. I will thank you for pointing it out. I will read it up.
I went down a rabbit hole. I started with Stuart Hameroff with the microtubule stuff
Yeah.
and anesthetics, and then it went
Yeah.
on to Roger Penrose and his ideas about consciousness.
Yeah. Okay. Well, thanks. I'll have a look at it.
Okay, another hand by Lawrence. Please go ahead.
So one application of quantum biology that I've been interested in over the years is the reaction mechanism of superoxide dismutase, which is the enzyme that protects against free radicals, right? And right, this is going to sound crazy, but you've heard of spontaneous human combustion?
Yeah, I used to be terrified of it when I was younger.
Really? Wow, that's a
Well,
talk.
well, it was.
But now let's see if we can combine two concepts of free radicals that are frustrated from being close enough to one another to quench each other, right? And so now you have these persistent free radicals, right? Now, what would be the thermodynamics of subsequent reactions that would lead to a like a combustion type reaction, and what would be a model system for that?
I can't imagine the recombination of radicals generating that much. I would see it as more a slow poisoning of your organs, or a slow organ failure, more than a combustion. I can't imagine it gives off that much energy, but I'm not actually 100% sure. So again, interesting question. I will go and look at it more. But as far as I know with radicals, is they just cause tissue damage or organ failure, or they basically are inflammatory. But I don't know how. I can't imagine they're inflammatory enough to make you burst into flames.
Right.
But, yeah. Thanks.
That would be a wild one.
Yeah.
Incredibly bad luck.
Yeah, exactly. It is, and it is a fascinating. It is. I'd completely forgotten that it was one of my childhood fears.
Okay, there's a question from Jebel Mahamud in the chat. He says it's a very fascinating topic. Currently, I'm working on the application of spectroscopic techniques for plant leaf disease detection. How would I probably apply quantum physics, and what software is suitable? And that's actually a question very well in the domain of my research.
Yes, I was going to say, Chart, do you want to go ahead? Yeah. Do you want to go ahead, because you're the experimentalist?
Right, Jebel, I would encourage you to email me, and I can send you some information on that. So there's not really commercial software available, and I think Bettany will agree with that. Normally, when you want to model something in quantum physics, you write your own code. So there are some software packages for certain things. I can send you some information about that. But let me put my email in the chat, and then you're welcome to email me. I will send you more information on that. Bettany, anything you would like to add?
That you also very well to that question. I know in terms of, because spin biology is my main field of research, and I know the mole spin is being developed by a group, and it can do pretty much everything with radical pairs. If you look up mole spin, it's a great package for that. I think it is in C. It's not in Python. But then there is also there is a Python package for spin radicals, I think. And then Qtip has some photosynthesis stuff as well. But again, let me put my, I can also put my email if anyone has further questions. I forgot to put it on. I didn't get to my end slide because I was so behind, sorry.
There are also a few people in the chat to ask whether you would be willing to make your slides available.
Yeah, absolutely. I'll send them to you, Chart, if that's...
We can do that,
Yeah.
but I would encourage everyone who's interested to maybe email you. So Bethany has put an email in the chat. If you're interested in the slides, then please contact her directly.
Yeah, perfect.
That will be much easier.
Yes, sorry.
Lawrence, see you have another hand.
Yes.
I was just going to build upon a comment that Sakazi was making in terms of the history. There was a guy named Elmer Imes, and his contribution to quantum theory was to prove that the quantum theory applied to molecules.
Okay.
So this was in the early 1920s.
Okay.
So that's another person to add.
I think, to be honest, the history of all, well, not only physics and biology, it's I mean, you look at you find some papers that were written in the 80s that were like saying the same things that are being written about now. It's just who ends up the figureheads of a scientific discipline aren't necessarily the only people who developed it. I think it's a bit unfair to have something like this timeline, but it was just to give an idea of some of the seminal ideas. But thank you for that, pointing that out. I'm going to go and look all those things up.
But you know, another experimental idea about this radical pair mechanism with respect to avian navigation is to, I mean, if you can ever get approval to do this type of experiment, is to put a bird in a magnetic field inside of a neutron beam. Right. Which is probably not great for the bird, but but I mean, it is an experiment where I think, I mean, the bird could survive the experiment. This probably wouldn't be the same afterwards.
Well, they do. I mean, there are quite good like there's quite good behavioral evidence of the radical pair. So, I mean, they do put birds into different magnetic fields and oscillating magnetic fields, which cause the birds to be, I mean, anthropogenic electromagnetic radiation of like radio frequencies from kilohertz to megahertz disrupts bird migration, which is a little bit disturbing. The nice thing is that it's a temporary effect. You remove them from it, and then they recover that sense.
Right.
But yeah, there's quite a lot of behavioral evidence for birds being disorientated by different magnetic fields.
So maybe this could be an experiment you could do with Safari right there in Pretoria.
Yeah, we're hoping to do some experiments with Chart, hopefully.
Right. Are there any other pressing questions?
You're very welcome to email me too. So.
Yeah, so we have gone a bit over time, but it's a fascinating topic, and thank you very much, Bethany, for
thank you very much for listening.
Some flavor of it.
Yeah.
So as Bethany has said, if you have more questions, please email her. You're also welcome to email me. I'm also in this field of quantum biology, much less than Bethany, but more from the photosynthesis point of view. But yeah, thanks again, Bethany. Thanks a lot for presenting today.
You. Thanks. Have a
thank
good day,
everyone
everyone.
for coming.
Bye.
Thank you. Goodbye.
Bye.
Bye.
Right.
Hi, Jart. How are you?
Hi, Raymond. Yes, it's nice to see you.
Yeah, and you. Yes. So I've been reading through the draft of the ASAFP report.
Are you asking what the status?
No. Trevor sent me a copy of it, so I'm just informing you that he sent me a copy, and I've been looking through it and just providing him with a little bit of a feedback.
Okay, right.
Oh.
Yeah, so if you have more feedback, you're most welcome. The process is quite slow. So
Yeah, I
they're
understand it's been back
organizing a
several years for it to
conference.
come. Yeah, it's been
Yeah,
several years to come to this point, yes.
yeah, exactly. Because I
Yeah.
already asked for inputs 3 or 4 years ago, and
Okay.
then I wrote this chapter 2 years ago, and yeah, now we are 2 years further, and they are still organizing a conference to discuss all the comments that I've received. That
Oh,
conference
okay.
would have taken place in September, but that has been postponed to November. So
Right.
it's
Okay.
a lengthy process. It just means that there's sufficient time for you to give your any
Right.
additional.
Oh, okay. Right. Yes.
Yeah.
Yeah. Would it be an online conference?
I'm hoping that they will make it a hybrid option. So they want
Okay.
to have in-person participation, but
Mhm.
I'm not able to go there physically. So hopefully they have this online option as well.
Okay. All right.
And the,
So,
yeah, so if there's a link available, then yeah, then I can also send it to you. As
yeah,
soon
if you
as I
could
know
please.
more, I will let you
Yeah.
know.
Yeah. I've sent the copy to Thomas as well because he was on the letter of support.
Okay.
Yeah.
Yeah, no problem.
Yeah. And just as a matter of just to keep you in the picture, because there was a section on physics education in it.
Okay.
And one of the constraints that was identified was in the